2020. This is our updated animal viruses PowerPoint, updated for some focus on the pandemic strain of coronavirus that's causing so much heartache right now. Okay, folks, so again, I'll try to break this into shorter audios because I know it's pretty dense information. Okay, so in our previous slides and previous audios, we had given a general introduction to viruses, and then we had focused on bacteriophage replication, the lytic and lysogenic cycle, and had talked about generalized and specialized transduction carried out by bacteriophage. So now what we're going to do is we're going to move from bacterial uh, phage biology into animal viruses. And when I say animal viruses, folks, I'm including humans. Very often, like in some readings, you'll see people describing humans and animals. So it's like we are animals, right? So when we say animal viruses, we're including viruses that can infect humans. And indeed, we'll see just with the novel coronavirus, many of these viruses are zoonotic. That is, they can be transferred from non-human animals into humans. And we'll see that with both the novel coronavirus that's causing the pandemic right now, and also with influenza viruses. So do you remember, you guys, the best description of viruses is they are trouble wrapped in protein. So why did our understanding of human and animal virus replication lag so far behind our understanding of bacteriophage replication? Well, for one thing, it's a lot harder to grow and study animal and human viruses compared to bacteriophage. And the problem is, remember that viruses are obligate intracellular parasites or pathogens. So in the early years of virology, to study these human or animal viruses, you'd actually have to inoculate infect animals or humans, and it remains maybe not a very ethical um, practice to do that, to potentially cause suffering in your um, animal subjects. So it was really good when it was discovered that some viruses, but not all, that's the problem. Some viruses can grow in embryonated or fertilized eggs. There's some disadvantages. One is it takes a lot of expertise to inoculate the eggs correctly. Certain viruses have to be inoculated into different parts of the egg, which we'll see in the next slide. And again, a big problem is not all viruses will grow in embryonated eggs. Um, one just kind of a connection here, you guys. So with our annual influenza vaccine, the vaccine viruses are grown in embryonated duck eggs. And that's one reason why if you go to get the influenza vaccine, they'll ask you if you have any allergies, especially to eggs, because they don't want the vaccine to trigger your egg allergy. Another problem with growing viruses in embryonated eggs is there's great potential for the eggs to become contaminated, and then you lose all of your vaccine viruses. And furthermore, for example, with the influenza vaccine viruses, it takes them about 6 months to grow enough influenza vaccine virus in eggs for the vaccine. So with with influenza vaccines, people have to guess 6 months in advance which types of influenza viruses to include in the vaccine. And some years they're wrong; they guess wrong. So maybe we'll have time to talk about that a little bit more. Okay, and then, folks, there was in the 1950s there was a kind of a breakthrough in that folks learned how to grow cells in what's called cell culture or tissue culture. So this was about in the 1950s. And this permitted folks to grow animal cells or human cells in the lab in sterile plates fed with a medium. And normal human or animal cells, they'll grow in monolayers, a single layer. In the old days, they were the sterile plastic tissue culture, what they called plates, functionally like a sterile petri dish. And then if you were lucky, if the particular virus you were studying, if you were lucky, you could find a tissue culture cell line in which your viruses could grow. So you could add your viruses of interest to the tissue culture, and hopefully the tissue culture cells would have would still express the right receptors so that your virus could bind and infect and replicate. And then they'd be released in huge amounts in these tissue cultures, right? So the viruses that could be grown in tissue culture, this was a huge advantage. It really helped folks understand the natural history, the biology of those human or animal viruses. It permitted folks to do antiviral drug studies using the virus tissue culture models. It permitted the growth of large numbers of viruses for vaccines. So, for example, the polio virus could be grown in tissue culture, and the viruses could be harvested and then used in polio vaccines. But again, you guys, were trying to update some of these slides using our coronavirus, our novel pandemic 2019-2020 coronavirus. A huge problem, you guys, is coronaviruses in general do not grow well in tissue cultures. So this means it's difficult to study all the details of the replication. Some of the details of the replication we have to base on replication of viruses that are similar to coronaviruses. In the case of human coronaviruses, sometimes we have to use other animal um, other animal coronaviruses as our models. Um, so we just want to remember this, folks, that there's still kind of a lot of mystery behind the human coronaviruses, and that might be one of the problems, why we can't understand how it's spreading so rapidly, or why is it that certain people are getting maybe sicker than others, and how is it that it seems the coronavirus is so easily transmitted. So this is just to show you folks one of the earlier ways to grow viruses without having to inoculate mature animals or humans. This is the embryonated, the fertilized egg. So here's the little embryo right here. And this is just showing you that different viruses need to be inoculated into very specific sites within the embryonated egg. So this takes a lot of expertise, right? And then furthermore, you guys, you can imagine that because you're piercing the shell to inoculate the viruses, it'd be so easy to introduce microbial contaminants. And so if you got bacterial contaminants or fungal contaminants, that would destroy your viruses that you were making. And furthermore, folks, really important, remember a lot of viruses won't grow in embryonated eggs. For examples, the um, pandemic coronavirus doesn't grow in embryonated eggs. And over here, folks, this is a modern update on those old sterile plastic tissue culture plates. So this is a plastic bag. It would have a monolayer, a single layer of cells growing in here. The red is the liquid food, the medium for the cells, and it usually has a pH indicator in here to let you know if you have maybe microbial contamination. And then you'll see ports here. So you can see a port here. We've got a hypodermic syringe. So this port would let you, for example, inoculate the tissue culture cells with your virus of interest. It would let you um, introduce potential antiviral drugs. So you could see if maybe some new antiviral drug X, if you inoculate it, does it inhibit, stop the replication of the viruses, right? And then furthermore, you guys, once the viruses have replicated in the tissue culture cells, they'll be released into this medium. So you could also harvest that liquid medium to isolate the viruses, for example, to do maybe electron microscope studies, or maybe you could harvest them to make a vaccine. And this, folks, this is not, don't worry about these, the text here. I pirated this off the internet, but just to give you an idea of how in the old days when we used these sterile plastic tissue culture plates. So again, it functions like the petri dish that we use for our agar plate. So you would take, like, as an example, you guys, maybe you could take a piece of liver from an animal and grind it up, homogenize it maybe with a little bit of the growth medium, and then you would place those cells into your sterile tissue culture plate with the medium, right? The food, you have to have that liquid food there. And then if you're fortunate, the cells will start to divide, right? So animal cells, remember, the eukaryotes, so they're going to divide by the process of mitosis and cytokinesis. And then eventually they would they would divide and form a single layer of cells here, a monolayer. And normal cells, folks, they have a phenomenon called contact inhibition. So once they touch one another, they touch the sides of the plate, they'll stop dividing, and that's what keeps them in this beautiful monolayer. Excuse me. And so it's once you have this nice monolayer of cells grown, you could add your viruses. Excuse me. So again, you could add your viruses to your tissue culture cells. If the tissue culture cells have the correct receptors to which the viruses can bind, the viruses will invade the cells and replicate within them. So again, this is a wonderful way to study viral replication. You can harvest the viruses to make a vaccine. And as we said, you guys, you could test the potential antiviral drugs to see if your drug will stop the virus replication. Just another thing, folks, up here, some tissue culture cells won't express the receptor they would normally express in vivo, like if they're in their living host. And I believe that's a problem when they were trying to develop the hepatitis virus vaccine. Liver cells, my understanding is human liver cells in tissue culture will not express the receptor for the hepatitis B virus. So that made developing the hepatitis B virus vaccine much more challenging because we couldn't grow them in tissue culture. Okay. But again, folks, coming back to our pandemic coronavirus, the human coronaviruses do not replicate well in cell culture. And so that's really hampered or slowed down our understanding of the details of their replication. It's certainly going to slow down antiviral drug studies, and certainly it's going to be make it a lot harder to develop a novel coronavirus vaccine. There's all kinds of cool new technologies, you guys, though, in vaccine development, and perhaps we could even introduce that as another possible um, bonus topic, is looking at the newer technologies that folks are using trying to develop a coronavirus vaccine. That would be really awesome. But folks, there even if you have viruses that will grow in tissue culture, in these cell cultures, there's a problem. And the problem is that with normal cells, normal healthy cells, they can only replicate a limited number of times. And the reason for this is, folks, and I should have added this in the PowerPoint slide here. Let me see if I can sneak that in. Right. Okay. So the reason for this, you guys, the reason that cells can only replicate a certain number of times is on our chromosomes. At the tips of our chromosomes, we have these DNA sequences called telomeres. And every time our chromosomes are replicating, the telomeres get shorter and shorter. So, so the telomeres shorten. And once they get so short that now you're starting to lose coding, DNA coding sequences, the cell can no longer divide. So, sorry, I'm putting this in here late, you guys. Shorten. And, okay, so when the telomeres shorten, eventually it will stop, eventually stops cell replication. Okay. And the reason this is a problem for eukaryotic cells and not bacteria is, remember, most eukaryotic cells have linear chromosomes. And remember, prokaryotes, bacteria, have circular chromosomes, so they don't have to worry about losing coding sequences every time their DNA, their chromosomes are replicated. Okay, so the point being, folks, that in normal healthy cells, they can only replicate, they can only divide a certain number of times. So let's just pretend you guys were looking at some cell and it can only replicate 30 times. So if you were using that cell in your tissue cultures, you can only pass cells like 30 times. So you'd only have like 30 chances, right, to study your viruses in that particular cell line. And at the end of that, the cell culture would die out. So this, again, it hampered studies of viral biology and pathogenesis and immunity and treatment. So what the virologists really wanted was what we would call an immortalized cell line. meaning cells that would divide and divide and divide and divide, and they would never stop dividing. So immortal or immortalized, you're going to live forever. And the only kind of cells, like for example in animals and humans, that can divide and divide and divide and divide and never stop, as far as I know. Now, folks in A&P, you yell at me if I'm wrong, but as far as I know, the only immortalized cells that are going to grow in humans or animals are going to be cancer cells, right? And that's such a bummer. So this is such a tragic story. So virologist scientists were seeking, these immortalized cells that could be used in tissue culture, and they would just keep dividing and dividing. So this is the really heartbreaking story of Henrietta Lacks. So Henrietta Lacks, she was a young mom. She had little kids. She was a wife, and at a very young age, she developed and suffered with cervical cancer. And the the reason we need to bring this up, you guys, is that it's so important that we're ethical, and we behave morally in medicine and science. So the story of Henrietta Lacks, it's a story where um, her rights, the patient rights, were violated, the family rights were violated, and people act really unethically. So we want to learn from history and not ever do this again. Okay. So when Henrietta Lacks, she was basically dying in the hospital from her cervical cancer. Um, the physicians, the scientists, they harvested some of her cervical cancer cells and introduced them into tissue culture. And this was what they were looking for. So Henrietta Lacks, her cervical cancer cells were truly immortal. They kept dividing and dividing and dividing, and they could move some of the cells to new tissue culture plates, and they kept dividing and dividing and dividing. And indeed, in labs around the world, even today, Henrietta Lacks's cervical cancer cells are still dividing, right? So they're truly immortal. Now, when I was in college, they wouldn't release the name of the patient from whom the cervical cancer cells were harvested. And so we were told simply they were called HeLa cells, and we were told that was for Helen Lane, and that was totally incorrect. So the victim, right, that unwillingly donated these cervical cancer cells, again, it was Henrietta Lacks, right? So this is just totally unethical, you guys. They didn't ask her permission. She didn't know they were harvesting the cells, and she didn't know they were going to make these immortalized cell lines, right? And she died shortly after in 1951, leaving her kids without a mom, and her husband without a wife and a mother. And so her family didn't know her cancer cells were harvested and were being used in research around the world. And these HeLa cells, Henrietta Lacks cells, have been used in so many important studies. They were used in polio vaccine studies. They've been used in HPV studies. In fact, someone got a Nobel Prize working with these cells, which is on the next slide. They've been used in HIV studies, and I think probably the list of experiments and studies that have been done with the HeLa cells, I bet you there would be pages of them. So, indeed, one of the people that profited greatly from the HeLa cells, he discovered that a human virus called the human papilloma virus, so-called wart viruses, a specific strain, HPV strain 18, this virus had inserted its DNA into the chromosome of the cervical cancer cells. So this was a huge discovery. And I think this was one of the first times that it was discovered that specific viruses could increase the risk in humans of specific cancers. So it's now been shown it was this HPV strain 18 DNA inserted into the good normal cervical cells that had caused them to become cancerous. That is, they could no longer control their division, right? So when you win Nobel Prizes, you get a lot of fame, you get a lot of money. And again, Lack's family, Mrs. Lack's family, whoops, sorry. They didn't know. Again, they didn't know that any of this was going on. They didn't know a Nobel Prize had been won using the mother, the wife cells, and certainly none of the Nobel Prize money was shared with them. So just kind of all the way around bad news. So again, folks, this was a big discovery showing that there are some human viruses that are what we call oncogenic. Oncogenic means they can increase the risk for cancer. And we will see later on that, yes, there are certain strains of HPV, the human papilloma virus, that can increase our risk for cervical cancer, cancer of the vulva, anal rectal cancer, cancer of the penis. And now we're starting to see an increase in cancers of the throat that are caused by these HPV strains 16 and 18. And generally, folks, we think of the HPV strain 16 and 18 being transmitted sexually or when we're being intimate with a partner. So it was a journalist by the name of Rebecca Skloot that finally revealed this story to the world and to Henrietta Lacks's family in this book called The Immortal Life of Henrietta Lacks. I haven't read the whole thing, but the parts I've read are just like heartbreaking. And I do believe a movie was made based on the book The Immortal Life of Henrietta Lacks. So, folks, what we'll do, that was kind of an introduction of why our understanding of human and animal virus replication and biology lag behind our understanding of bacterial viruses. So what we'll next do is we'll start looking at some of the basic steps in human and animal virus replication. So these slides, you guys, I know they're really intense. And so with the next audio, I'll try to, like, maybe with each slide, I'll say, okay, this is what you need to take away from this slide. Like, this is what we could ask you on the lecture exam. Okay, folks, so we'll close this one here. We'll call this animal viruses, maybe part 1, and then we'll follow up with a few more audios.